Hello there, welcome to my presentation on a little process aptly known as clonal processing, involving the proliferation and differentiation of immunocompetent lymphocytes. The cloning of these cells occurs daily as part of our adaptive immunity each time a foreign antigen enters the body. An antigen-presenting cell, or APC, could be a macrophage, B cell, or dendritic cell, which has undergone the exogenous processing of a foreign antigen found outside cells in the fluid. They range from bacteria to parasites and to viruses yet to infect a cell. Once antigenic proteins are broken down into peptide fragments and associated with MHC2, the antigen MHC2 complex is inserted into the plasma, ready to be presented to a helper T cell. This inactive helper T cell has a specific receptor for this antigen and will only recognize the MHC2 complex presented. The surface protein CD4 interacts with the MHC2 antigen complex as a co-receptor during the binding process. Once this T cell is activated, it undergoes the most important process of clonal selection. First, it will proliferate and divide several times. Then it will differentiate and form highly specialized cells. Most will become effector cells, or clones of the helper T cell, while less will become memory helper T cells, which remain inactive until the next time the same antigen enters the body, ready to offer a prompt response. These helper T cells are so aptly named because once cloned, they then secrete different cytokines, which are small protein hormones that stimulate or inhibit many normal cell functions and aid in the adaptive immune response of the body. The cytokine interleukin 2, which is secreted, enhances activation and proliferation of cytotoxic and helper T cells, B cells, and natural killer cells, and it aids in the activation of cytotoxic T cells via a process called co-stimulation. The activation of cytotoxic T cells is very similar to helper T cells, the only differences being that the surface protein is CD8, not CD4, and that it interacts with the MHC1 and not 2 complex of an infected body cell, which has undergone endogenous antigen processing. Activation can only occur if a second signal is received during the binding of the cytotoxic T cell to the antigen via co-stimulation by the interleukin 2. The clones of the cytotoxic T cells are seen as soldiers in the battle of immunity, as they will continue to attack the infected body cells until the immune response has completed. So overall, we can see that both of these processes allow the body a fighting chance against any foreign invaders. Before clonal selection occurs, there is only a few lymphocytes able to recognize the specific antigen. But once it occurs, there are thousands, an army of highly specialized lymphocytes ready to fight off any foreign invader.